And so now we're going to learn about non-Mendelian inheritance, or some of the inheritance patterns that he did not talk about. One of them is called incomplete dominance. So as you can remember from the law of dominance, we essentially said that the heterozygote will always have the same phenotype or appearance trait as the homozygous dominant flower. But as you can see here, in incomplete dominance, the heterozygotes do not display the dominant phenotype. We would expect normally that the heterozygotes would all be red because of the law of dominance. But here they're actually pink. And so this is a snapdragon. This is what they usually use to demonstrate the law of, not the law, just incomplete dominance in general. And so in incomplete dominance, the heterozygotes don't completely follow the law of dominance. They do not show the dominant trait. Instead, they show their own unique heterozygote trait, which is intermediate between the two dominant and recessive phenotypes. So the dominant phenotype is a red flower. The recessive phenotype is a white flower. And as you can see here, the heterozygotes have pink flowers. So red and white, and in between we have pink. The way you should actually think about this, though, is just that the red color is being expressed, just not to the same degree as if everything was dominant. So it's just a bit of red, so it's pink. But the easy way you can find out if it's something's incomplete dominance, or the way I like to think about it, is if the phenotype is intermediate between the two dominant and recessive phenotypes. So it's a blend. So the genotype frequency now is equal to the phenotype frequency, 1 to 2 to 1. And again, this is because now the heterozygotes have a unique phenotype. And so the phenotypic frequency is 1 to 2 to 1 instead of the homozygous dominant and heterozygous being added together to make 3 to 1 like in our monohybrid cross. So genotype frequency equals phenotype frequency is equal to 1 to 2 to 1. Red, pink, white. So that's incomplete dominance. Remember this very closely. It's an intermediate phenotype. Because we're going to talk about something that a lot of students get confused with next, which is codominance. So as you can see, both of them have the word dominant in their names, and sometimes it's really hard to separate them. In codominance, you have 2 or more alleles that are codominant with each other, and they're both expressed. So normally I would have, if I'm looking at the blood type system, right? And if I was Gregor Mendel, I would say, for example, that, oh, this A allele is probably dominant to the B allele. So if I had a genotype of AB, I would expect to only have a blood type of A because A is dominant to B. But that's not actually the case. In codominance, they're both dominant and they're both expressed together if it's heterozygous. So if I have type A, if I have these 2 A alleles, then I have type A blood. If I have these 2 B alleles, I have type B blood. But if I have the A allele and the B allele, I actually have both. I am a type AB blood. I show both of the phenotypes A and B. So that's codominance. You have both of them. You have both of these phenotypes expressed. Whereas in incomplete dominance, you have a blend of them expressed. And so, for example, if this flower was codominant instead of incomplete dominant, you would actually see maybe half the petals would be red and half the petals would be white because both of the traits are being shown. But this is incomplete dominance. So they're being blended together in incomplete dominance, and they're both being expressed in codominance. And the ABO blood system is actually a very good indication of another non-Mendelian inheritance pattern, which is multiple alleles. As you can remember, Gregor Mendel said, life is easy. We only have 2 alleles. We have a dominant allele and we have a recessive allele. But sometimes there's more than 2 alleles. Again, in this ABO blood system, we have an A allele, we have a B allele, and we actually have another allele that's neither A nor B. This one's actually the recessive allele. So A and B are co-dominant with each other. They're both dominant to this recessive O. And so what you can see here is the different types of blood types we get. So if you have the genotype of AA, you have, again, that's homozygous dominant. So you have the blood type A. If you have AO, A is dominant to O, so you have the blood type A. Same thing for type B, right? You have homozygous dominant, and you have B is dominant to the O, so you have type B blood. Type AB is codominance, where you have both the A and the B traits being expressed on your blood cells. And then in type O, which is the recessive genotype and phenotype, you don't have either A or B. And so as you can see, just like we saw with Mendel, the recessive phenotype is only shown when it's homozygous recessive, when both of the alleles are recessive. Otherwise, it's going to be covered up, dominated, masked by the dominant allele. Here's another interesting one that they like to talk about a lot in non-Mendelian inheritance, epistasis and polygenic traits. So in epistasis, the interaction between 2 or more genes control a single phenotype. And so I borrowed this illustration here to kind of illustrate what epistasis looks like, because this definition looks really confusing. Interaction between 2 or more genes controlling a single phenotype. So let's say that there's a gene that controls what color your hair is, right? It's going to be blonde or red, or in my case, my hair is black. And then also there's a separate gene that controls baldness. So if you're bald, you have no hair. And so this is epistasis because your hair color, actually this trait, like what your hair looks like, really depends on whether you're bald or not. And so if you have this blonde hair, you look blonde. If you have red hair gene, then you look like you have red hair. But if you have the gene for baldness, it doesn't matter because you don't have hair anyways. So this gene that's controlling what color your hair looks like doesn't matter anymore. There's an interaction between 2 or more genes that are controlling a single phenotype, and that's what your hair looks like. So you have the gene for the hair color and the gene for baldness. They're interacting together, and together they decide what your hair is actually going to look like. Because if you're bald, you're not going to have hair color anyways. Multifactorial polygenic inheritance is true for most of our traits, actually. There's no single gene for something like height or skin color that's just like either like a tall gene or a short gene. We're not pea plants. It's more complicated than that for humans, for example. There's a wide variety of different genes that are all interacting together. For example, for skin color, I think there are 4 different genes that are determining your skin color. Now we're going to try to tie what we learned from Mendel and classical genetics into molecular genetics. So you might have heard of DNA before, and DNA is actually bundled up into these chromosomes. You might have heard of these 23 chromosomes from the company 23andMe. So humans have 23 pairs of chromosomes. Genes are actually located on these chromosomes. Like you would be able to locate their physical location on this chromosome, and these genes will code for proteins. So this red hair gene is on this chromosome, like for example, chromosome number 2. And it codes for this protein that makes your hair red. And so the first 22 pairs in humans are called the autosomes. These are like your body chromosomes, and they're inherited 1 from your father, 1 from your mother. That's why you have 2. That's why they're in pairs. And this is called a diploid because you have 2 sets of each chromosome. And this explains why there's 2 alleles in the gene normally, because you have 1 variant from your father on 1 chromosome and 1 variant from your mother on another chromosome. There's also a 23rd pair, and those are your sex chromosomes. So these are actually different depending on which sex you are. So if you're male, you have X and Y chromosomes, so this is a male. And if you're female, you only have an X chromosome. You have 2 of these X chromosomes. And some chromosomal disorders you might want to know about are trisomy, which means you have 3 chromosomes. And the most famous example is trisomy 21 would give you Down syndrome. So that would have 3 chromosome 21s. And monosomy, which is your missing chromosome. So instead of having 2, you only have 1 for a certain pair. So instead of having 2 X chromosomes, if you're a female, you only have 1 X chromosome. That's called monosomy X. That's called Turner syndrome. And so what we just saw here is that genetically, chromosomally, males and females are different. And this leads to another non-Mendelian pattern of inheritance called sex-linked inheritance, where the expression is dependent on the sex of the individual. So you might have known about the royal families in Europe. They were pretty inbred. They married off a lot to their cousins. There was a lot of family marriages going on here. And so what they suffered from is a disease called hemophilia. And hemophilia, when you get cut, your blood is not, you're not clotting as fast as a normal human would. And so you tend to bleed out. And so the European royal family in the 1800s and 1900s, they tend to have this problem. All these red people that are outlined in red here had hemophilia. And you might notice something interesting. I think all of them are male. The females, Queen Victoria, who was a carrier for hemophilia, her daughters, her granddaughters, none of them suffered from hemophilia. But her male line descendants tended to suffer from hemophilia here. So her male descendants were at this hemophiliac risk. And this is because males only have 1 X chromosome. So if the trait for hemophilia, this gene, is inherited on the X chromosome, and it's like X-linked recessive, right? Normally we would say the law of dominance says I have 2 alleles. If one of them is dominant, if I have like a hemophilia gene that's normal on 1 chromosome and a hemophilia gene that's diseased on another chromosome, the one that's normal will dominate over the recessive allele that causes hemophilia. And this is true for females because they have 2 X chromosomes, right? So if you have 1 diseased X chromosome, fear not, because you have 1 that's healthy usually, and that'll prevent you from showing the symptoms of the disease. But if you're male, you only have 1 X chromosome because your other chromosome is coming from your father. And so if this X chromosome is diseased, you have no dominance that's going to cover up the hemophiliac trait. You're going to show as a hemophiliac. And so examples of sex-linked inheritance traits are usually red-green color blindness. You might know that that happens to males much more often than females, and same for hemophilia. So yes, males only have 1 X chromosome, and if they have a recessive allele for a gene on the X chromosome, like the hemophiliac gene, it will be expressed. And as you see, here's the Punnett square cross. For sex chromosomes, you have X chromosomes from your mother, but for your father, you actually have just X and Y. And so you have a risk of showing this hemophiliac trait if your mother is a carrier. So we just talked about sex-linked inheritance. There's actually 4 general types of inheritance that you should know. Autosomal dominant, so there's a karyotype 22 autosomes. We talked about these 22 are your autosomes. If it's inherited on your autosomes, then it's autosomal. If it's inherited on your X chromosome, it's X-linked. And so dominant recessive tells you whether the allele for the disease is dominant or recessive. So again, if it's dominant, it will show if you have a dominant allele, 1 dominant allele or 2 dominant alleles. And if it's recessive, you have to have both alleles to show the disease. So an autosomal dominant disease is polydactyly, which is having, poly means more than, and dactyly refers to your hands. You have more than 1 finger, more than 5 fingers on your hand. So usually you'll have like a 6th finger. You can also have multiple toes, more than 5 as well. That's polydactyly. It's an autosomal dominant trait. Autosomal recessive trait can be Tay-Sachs disease, which is a degenerative disease of the brain and the spine resulting from lipid excess. X-linked dominant diseases are very rare. Rett syndrome is one of them. And X-linked recessive diseases tend to affect males. So hemophilia, red-green color blindness, these are all examples of X-linked recessive diseases. And now we're gonna have another poll just to